Ever wondered how DNA fingerprinting works? It's a fascinating process that makes use of unique patterns in our DNA. This intriguing science is critical in areas like forensic investigation, paternity testing, and genetic research. Two primary methods are commonly used: Restriction Fragment Length Polymorphism, known as RFLP, and Polymerase Chain Reaction Amplification, often referred to as PCR. Each method has its unique set of steps and principles. Stay tuned as we delve into the first method, Restriction Fragment Length Polymorphism, or RFLP. Restriction Fragment Length Polymorphism, or RFLP, is a technique that scientists use to analyze variations in DNA sequences. Let's delve into the fascinating process of RFLP and how it aids in creating unique DNA fingerprints. Our journey begins with the isolation of DNA from the sample material. The sample size for an RFLP test must be substantial enough to yield accurate results. Once we have the requisite quantity, we separate the DNA from the sample, preparing it for the next step: restriction digestion. In this process, we introduce restriction enzymes into the isolated DNA, which act as molecular scissors, cutting the DNA at specific locations. The cut DNA, now a digest, is separated based on size using a method known as agarose gel electrophoresis. If you think of the gel as a maze, the smaller DNA fragments will travel further through it, while the larger ones lag behind. This separation creates a unique pattern, much like a barcode, representing the DNA sample. Next, we transfer the separated DNA from the gel slab onto a nitrocellulose membrane, a process known as southern blotting. The membrane is then exposed to a labeled probe that's specific for one variable number tandem repeat, or VNTR region. This probe is essentially a complementary sequence for the VNTR region nucleotide sequence, and it's labeled with radioactivity to make it detectable. Following the hybridization with the radioactive probes, we develop an X-ray film from the southern blot. Only the areas where the radioactive probe binds will show up on the film, appearing as bands. These bands represent different VNTR regions in the DNA sample. Finally, we compare these bands with other known samples. The pattern of bands, or the DNA fingerprint, is unique for every individual, making it an invaluable tool in fields ranging from forensics to paternity testing. With RFLP, scientists can compare DNA samples and create unique DNA fingerprints. This method, though labor-intensive and time-consuming, provides a high degree of accuracy, making it a cornerstone in the world of genetic analysis. While RFLP is a powerful tool, it comes with its own set of advantages and limitations. One of the major advantages of RFLP is its high degree of accuracy. It's often considered more precise than PCR, largely due to the larger sample size used and the use of fresh DNA samples. This reduces the risk of amplification contamination, enhancing the reliability of the results. However, RFLP isn't without its drawbacks. The process can be time-consuming, often requiring a longer duration to complete the analysis compared to other methods. In addition, the cost associated with RFLP can be significant, making it a less accessible option for some. Despite these limitations, RFLP remains a crucial method for DNA fingerprinting. Now let's turn our attention to another widely used method in DNA fingerprinting: Polymerase Chain Reaction, or PCR. This ingenious technique allows us to amplify a single or few copies of a specific DNA segment, creating thousands to millions of copies. In the realm of DNA fingerprinting, PCR is a game changer, enabling scientists to analyze even the smallest samples of DNA. It's like having a high-powered magnifying glass that can zoom into specific parts of the genetic code. But it's not just about quantity. PCR also offers high-quality, reliable results in a shorter time frame compared to other techniques. Intriguing, isn't it? In our next video, we'll delve deeper into the fascinating world of PCR. We've come a long way in understanding the methods of DNA fingerprinting. To recap, we delved into the intricate process of Restriction Fragment Length Polymorphism, or RFLP, a method which involves isolating DNA, digesting it with restriction enzymes, and separating it based on size through agarose gel electrophoresis. We learned about southern blotting, which transfers and hybridizes DNA onto a nitrocellulose membrane to interact with a specific VNTR region. The final result of DNA fingerprinting is achieved by comparing these bands with known samples. Despite RFLP's high accuracy, we acknowledged its limitations: the need for a large sample size, fresh DNA, and ample time for completion, making it a costlier method. We also introduced you to another method, the polymerase chain reaction, or PCR, which will be our focus in the next video. Stay curious, keep learning, and we'll see you in our next video where we unravel the mysteries of PCR.